Welcome, everyone. Thank you for joining us for today's webinar, Tools and Processes for Making Evidence-Based Policy Decisions, presented by Eugean Jiwanmall, Senior Research Analyst for Medical Policy and Technology Evaluation of Independence Blue Cross. My name is Xavier Gutierrez, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. And if you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow your process to obtain your credits. Now, without further ado, please join me in welcoming Eugean Jiwanmall. I will now turn the presentation over to him.
Thank you, Xavier. So thank you for the introduction. Yes, so a lot to get through today with this presentation. As the title states, there are multiple tools and there are multiple processes. But one thing I always like to start off with is my key, and that's also the key for this presentation. That key being evidence. So we're going to revolve around a lot of as the title says, tools and processes, and we're going to go through some of them in great detail to understand how policy positions, reimbursement positions, in general, decision making is performed for any sort of technology that is out there, medical services, clinical services. But today we're going to focus in on molecular diagnostics. So for this particular presentation, I thought that because given the current environment, what sort of molecular diagnostic realm would be perfect for the audience in order to gain the most knowledge in the time frame that we have. So I figured that, let's talk about genetic testing as a base. I'm going to use that as an example, but the tools and the processes and the intricacies related with them apply across the board for any sort of molecular diagnostic decision-making regarding technologies that we'll be talking about, keeping evidence as the key perspective. So for genetic testing, there are multiple uses. I've listed a number of examples here. Diagnostic testing, predictive, being prognostic, being part of it. Your prenatal testing, non-invasive prenatal testing has been a hot topic for some years. Screening, and then, one of the favorites for many in the industry as well, like academia, pharmacogenetic testing for therapeutic purposes, for monitoring purposes. So again, just to highlight that there's multiple usages, and what we will be talking about today of how to go about evaluating the evidence for these type of testings. There's uniformity. Of course, there's going to be some specificities that we would have to consider as we go along, but I want to provide general frameworks which can be used for all these different varieties of testing and technologies that are out there. So just wanted to, since I gave you background regarding multiple different fields that our frameworks that we'll be talking about can apply to, let's try to dwindle it down and talk about how the questioning may start, as the slide says. So we can divide up the testing realm, the lab realm, into diagnostic, prognostic, and therapeutic. Again, the general questions would remain the same. That we'll be talking about and going over. How useful is the test? Is the test up to par? Is it doing what it's supposed to do? But if you wanted to make your questioning narrower, you can decide if it's a diagnostic, as a prognostic, or therapeutic. And then on the right side, you will see of the slide, you will see that you can even get further into more details. What is your patient population. Are you dealing with symptomatic individuals? Are you dealing with asymptomatic individuals? Are you doing testing where you need to know the history of family members as well? And then going down even further in the symptomatic population, are you talking about germline variation? Are you talking about somatic variation? Within the variation, are you dealing with benign versus likely pathogenic versus pathogenic variation, or are you working within variation of unknown? So there's a number of questions that start, and it's really important to have those drilled down before you get into your evaluation pieces, because that's what's going to decide further questioning and also the sort of evidence and that you'll be looking for and selecting, and then making an eventual decision based on how good that evidence is. Is it sufficient? Is it of high quality or not? We will talk about those points a little later on. And then also, under the asymptomatic population, I have screening, and DTC stands for direct to consumer, what kind of testing it is. So I just wanted to provide some examples in that sort of realm as well. And one of the other things that we have to keep track of it is the regulatory environment. When it comes to genetic testing, FDA is feeling its way out. They're getting more involved over the past few years. We have the in the kit world, because it's designated as a device, FDA approval is required. Then I have examples of companion diagnostics and complementary diagnostics. So for companion diagnostics, some of you may have heard of examples regarding some tests where FDA and CMS, which is they have partnered to do parallel reviews in order to understand and come up with positioning regarding companion diagnostics, which would be relevant for your pharmacogenetic tests that are out there. And so you have examples where there's regulatory bodies, and this is specific to the US, but it's across the board in the UK and other European countries and Asian countries as well. And just wanted to highlight in this slide that if a particular testing or diagnostic piece, the therapeutic piece or prognostic lab requires regulatory approval, it's really important to study that and understand it and see what the approval is for. And then within USA, you obviously have for laboratory development, developed tests, CLIA approvals. If you guys have more questions, you're more than welcome to share them with me, and I'll try my best to explain this particular field. It's murky at this time, how it applies to genetic testing, for instance, specifically, but I can answer those questions. All right, so let's get into the decision-making frameworks. What I won't be discussing today is USPSTF, EGAP, and GTT. Just wanted to list them there to state that there are more examples of other than what I'm going to be talking about. So maybe in a future presentation, I'll go over them. However, I decided to today talk about 5 criteria. I'll get into what that means. The ACCE model, and then the PICOTS model. So let's talk about the criteria, right? So some folks are familiar with BCBSA, which stands for Blue Cross Blue Shield Association. Their tech criteria is the technology assessment. It's the analytical framework of how to critically analyze and ask the correct questions regarding any sort of clinical medical service. And when I say technology, that's what I mean, medical clinical service, including laboratory testing. It's a framework which is extremely useful, used by multiple organizations over the years, and something that can be extremely useful even when you're developing the sort of evidence for the testing that you're tasked with developing and then bringing to the market and then eventually bring to the clinical world. And if you're in the business of evaluating evidence to come up with a policy position, this is a great framework. So there's multiple questions that you ask. We talked about number 1, the technology must have final approval from the appropriate government regulatory bodies. I talked about that in a couple of slides ago. So so just pay attention to that. If it is supposed to have a regulatory approval, does it have it or not? The second one is second point is does scientific evidence must permit conclusions concerning the effect of the technology on health outcomes. So that's where we get into what sort of study designs are required, what sort of sample sizes, what kind of statistical analyses are required in order for you to reach a conclusion that the test is clinically valid or useful. It's performing the services it's pitching that it is actually doing. So the peer-reviewed evidence, the publications must permit those conclusions. You need to have those conclusions. There shouldn't be any gray area. If there's any limitations or biases involved in the overall aggregate evidence that you're dealing with for the particular lab service that you're evaluating, then basically the evidence is not letting you make definitive conclusions. And you're in the sort of experimental investigational further studies are needed to answer the outstanding question, arena for the particular questioning that you're doing. The third point is technology must improve the net outcomes. So we can get more in detail about that, but just for now, the understanding of this is that the benefits that you're getting, clinical benefits that you're getting from the service that you're evaluating, it should those benefits should outweigh the risks. The fourth point, the technology must be as beneficial as any established alternatives. That alternative can be doing nothing currently. That's the traditional way of dealing with the indication or the indication that we're talking about. Or it could be that there are other methods, other lab tests, other monitoring services that are available. Whatever you're evaluating, be it a battery of tests or a singular test or a panel test, the results of those and the clinical outcomes that that are the primary clinical outcomes that you're achieving through that testing should be clinically equivalent or superior to the established way or the traditional way or the what is part of routine clinical care right now in the clinical community. And that sort of leads us to the fifth point, which is the improvement must be attainable outside of the investigation stage. What that means is that in the simplest term, that whatever you're bringing to the marketplace or you're introducing, it has to become part of routine clinical care. So the average patient which fits the criteria for whatever testing is being performed, the physician group or the healthcare provider is employing that testing across the board for every patient that fits that particular criteria. So basically routine clinical care is equivalent to that. So the point I would like to read that I think is extremely important. So at the end of the day, the evidence reviews assess whether a medical test is clinically useful. A useful test provides information to make a clinical management decision that improves the net health outcome. So a lot of buzzwords there. Clinically useful, the testing has to be clinically useful. The results that you get from it should have direct patient management impacts. And you need to be improving the net health outcomes that are desired. So at the end of the day, the next three lines, I think I've gone over it. But if you take the quotations and make that the goal during your evidence development of what sort of studies and what sort of evidence you need to be publishing for your labs to eventually get reimbursed or to have medical necessity positions for them, those are keys to keep in mind. So let's get into CDC stands for Center of Disease Control, the ACCE model. The slide you guys have, I will give you a couple of seconds to sort of have it at your leisure. But I try to provide references across the board. They're extremely useful. And if you have any questions or need further references or these references, more than welcome to reach out to me through LabRoots. So the ACCE stands for analytic validity. I introduced technical validity at that Point two, the first C stands for clinical validity. The second C stands for clinical utility. A lot to say there. And then the E stands for any ethical concerns or considerations that need to be taken into account. This is another model, and I think majority of the audience is going to be familiar with this, but I'll go into a bit more detail about it because this is the key framework that comes during discussions in order to make any sort of evidence policy decisions or to evaluate if the testing is ready for prime time routine clinical care, the points that I've talked about before. There's a lot of questioning that goes into it. Again, I would encourage you to turn to the references that I have listed, but let's get into a brief synopsis of what at least the ACCE is. So like I said, A stands for analytic validity, the technical validity. To summarize what that is your test accurate or precise? The technical sensitivity and specificity numbers. Is it picking up what it's supposed to do? So in the genetic testing world, is it picking up the variant that it's claiming to pick up, or multiple variants, depending on the testing that you're talking about? So take-home message is the test accurate or precise? And I get into more detail in the slide. But again, I like to bring out the buzzwords when there's a lot on the slide. Is it accurate and reliable? So when you keep repeating it, testing, is it going to pick up the variants or whatever, mutation or mutations that it's stating that it will within gene markers or any sort of markers that are out there. The second point is clinical validity, CV. Again, I'm sure that multiple of the audience have heard this before, but how I like to explain this is the genotype to phenotype correlation that you're speaking for genetic tests that are out there. Is there biological relevance? And for this, it would be before the testing is being developed or when it's in the sort of conceptual phase. These are the questions that would be asked. Does it make biological sense? If you're dealing with one marker or multiple markers, clinical validity for all of those needs to be established. And here's where the clinical sensitivity. So in the previous slide, I was talking about technical sensitivity and specificity. In this particular, for this particular requirement, you'll be looking at things such as clinical sensitivity and specificity, your NPV, PPV numbers, negative predictive values, positive predictive values, and the evidence and the level of evidence that went behind generating those numbers. These are all part of the evidence reviews in order to come up to see if the testing is eventually going to become part of routine clinical care. Is it medically necessary or not? And this is a very prime and major point of evaluation as you go through the evidence for any sort of test or technology that you're evaluating. Then our friend, the biggest requirement there is clinical utility. Now, like I said before I get into details about this, like I said before, there are many different types of testing that are out there. And then even when you try to reduce them to the diagnostic, prognostic, therapeutic world, it's worlds. Still, there's going to be variation, but the common theme is these things. So for instance, for clinical utility, you may ask several very specific targeted questions depending on what you're evaluating. But at the end of the day, clinical utility is that the evidence has to show that the results that you're getting from your testing are having direct patient impact on direct patient management. So in the diagnostic world, is it diagnosing correctly and then it's leading to the correct selection of whatever? Is it monitoring next? Are we going to be doing a referral? Whatever there is having a direct impact on it, it's leading you towards that. And in the therapeutic world, that's an example that is easier to understand. The testing that you're performing, are they helping you select the correct, accurate therapeutic option or options that are there, or are they telling you what not to use as a therapy? So clinical utility results and evidence behind it to support the quality and quantity of that evidence becomes extremely important. And this is where, a lot of the testing that is in the marketplace, this is a prime requirement before reaching a medical necessity level. And then obviously you have, ethical, legal, and social implications. And again, I would encourage you to, look at the references and study more about it. Just for the sake of time, I won't spend that much time on it. The next figure, I am sure that 99.9% of the audience has seen it, either during their academic, experiences or as they move to industry or their professional world. This is hierarchy of evidence. You will see many different variations of this particular pyramid or how it's explained. But this lays the groundwork of sort of the quality that goes behind it. So I talked about a lot of the measurements and a lot of the requirements behind the evidence, but this shows you that as you go up this pyramid of how the quality increases. And obviously, it's not stated here, but in order to move up this pyramid, it becomes harder and harder from a logistical perspective too. But this is sort of to give the idea that, why just having a case report or multiple case report, a case series is likely not going to be enough to establish clinical utility, why you need to move into control studies and if possible into randomized control studies, because you're reducing the biases and moving up, and you're able to pinpoint exactly that the outcomes that are being reported are actually you can trace them back to the sort of intervention or the testing that we're talking about. So again, you will see different variations of this, but this is sort of how the evidence progresses when it comes to the quality piece of it. And then I'll talk about the quantity. You can't let go of the importance of quantity when you're taking a look at it, but it varies again depending on the question that you ask. So let's get into the analytic or technical validity. What kind of evidence are we talking about? The slide discusses the higher confidence in order to establish analytical technical validity would come if you have multiple sites participating. There are multiple locations or labs or groups that have performed the testing in order to validate it. There's a large number of samples that were involved. There's peer-reviewed publications of that number. So what you see is a lot of times this data is proprietary, but if you publish it and it's available, it has gone through the peer review process, that gives you additional credibility to establish that accuracy and precision that you need to in order to establish the analytic or technical validity. And then, if you have regulatory body involvement and they give it a check for you, that's also a big positive. So in the next generation sequencing world, the platforms that are involved, you will see that at this point, if a test is employing a next generation sequencing methodology, for those platforms, unless there are glaring questions, it's in 2019 it's taken that the platforms have technical or analytic validity established, and you would move on to the next step, which would be clinical validity. So this I would encourage the audience to definitely. The paper is a couple of years old or less than 2 years old, but it's still relevant. That reference, I really like it. I try to use it a lot during my presentations. From this particular reference, I describe how the level of clinical validity when it comes to evidence is established. The good news here is you don't need controlled or randomized studies in order to establish clinical validity for whatever testing you're bringing to the marketplace. If you see on the slide, independently cohort, case control, or family linkage studies may be enough to establish the genotype to phenotype correlation that you're trying to do. But this is I would take like to take a pause here and spend some more time on it. So when to talk about panel testing, or when you're testing for simultaneously testing for multiple markers at the same time in your testing, what happens is that let's say you're looking at 18 markers of different kinds. There may be clinical validity, the genotype to phenotype correlation, if your markers are genes, for instance, for a couple or one or 2 to 3. But what about the rest of the 15 or 16 or 17 markers in my example of the 18 marker panel? You need to have clinical validity for as many, or preferably all of the markers that make up your testing. So that point is extremely important. But this slide discusses with you of so this is in the development of testing. This is where you would just use peer-reviewed literature that has been published in order to come up with the markers, and that can serve as when you're getting questions regarding your clinical validity by any organization. Those are the submissions that you would like to make. But again, yes, you can use observational studies, cohorts. And retrospective analyses are perfectly fine, depending on the situation. But they still have to be the quantity may have to be large depending on the testing that you bring into the market. So it may not be a one cohort study that's enough. It may be 2 to 5 studies with enough large sample size in order to have the confidence in establishing clinical validity. Again, there's going to be variations. If you're talking about a rare disease, obviously the prevalence numbers are not there, so the sample size asked is not going to be as large. But if you're dealing with a chronic condition that can have a large patient population, obviously the sample sizes required are going to be larger. So just keep those points in mind. And then our friend clinical utility. And here's a lot of, again, I don't have time to today to talk about, direct versus indirect evidence that can establish clinical utility. So I'm going to focus on direct evidence. And you see that obviously the best way currently to establish clinical utility would be do well-designed and independently replicated RCTs, randomized controlled trials. Now, this is where a lot of impasse occurs between organizations and the folks who are trying to get reimbursed or trying to establish clinical utility or medical necessity for their testing, that RCTs are not feasible, or the best way to go about it. Why can't we go? But that's a discussion for another presentation, and I hope I get to discuss that in the next year or years to come. But for now, this is a good primer to have to see how, in order to establish clinical utility for your testing or technology that you're bringing the marketplace or are utilizing or developing or having discussions about, this is the kind of data. And again, what is clinical utility going to establish? Is going to showcase that the results that you're obtaining from your technology or results or tests are having a direct impact on whatever patient management that is desired. At the end of the day, what would the criteria look like? Again, the first bullet is, explains itself. The second, third, and fourth bullet are what we just talked about, that the test is has the technical numbers, the technical sensitivity and specificity. It's accurate and precise. It has been established. The third bullet point on the slide is what I've been talking about, the clinical validity. It's genotype to phenotype correlation based on the evidence that the correct quality and quantity of evidence showcases that the clinical validity is there for the testing. All the markers, if you have multiple markers in your testing, it has been established for those. And again, I say that with the disclaimer that while you have discussions, it may be that a sufficient number of markers have it. Because we're at that point now where obviously if you have a 300+ marker panel test, highly unlikely, and I would like to say almost 100%, I can say with confidence that the clinical validity for all 300 of those markers has not been established. But that's a discussion. And again, you can ask in your questions about that. I can explain further more of how that discussion may go and what to do in those situations. And then the last bullet, extremely important. This is where a lot of the conflicts happen, unfortunately, because the evidence is missing for the clinical utility piece. And I'll repeat it, that peer-reviewed, not anecdotal, not posters, not abstracts, peer-reviewed data, published data establishes the objective effects of the test results on direct management and care of the individual for all applicable indications and clinical situations to verify clinical utility of the test. So a lot of buzzwords there. If you're dealing with multiple indications, the test has to perform well across the board. Results have to have a direct correlation and direct impact on patient management, whatever the poor clinical protocols are in place. So again, clinical utility extremely important. But for the sake of time, I'll move on. But this slide is extremely important. This is how if you have a medically necessary test, this is how the criteria would be laid out for it. I would skip over this slide and come back to it. It's the PICOTS model, which I will explain in later slide what it means. But this is a good example of how to go about it. I included it in here because I wanted to share the reference with you folks. Excuse me. So I encourage. I'm really big on references and going back and for my audience to go back and study them in greater detail, because obviously these are huge topics, and a 45-minute or even an hour presentation is probably not good enough to get the gist of it. So please go to this particular reference. I would highly encourage it, and any other ones that I have cited. There's a reason I cite them, obviously, because I want you guys to go and read further on it. All right, so let's get into the PICOTS model. So what have we talked about so far? We have talked about the 5-step criteria. We have talked about the ACCE model, but more and more you also see PICOTS, PICOTS. And again, I encourage to go to the FDA and AHRQ. I'm using them as a base to explain this particular model, which can be used for evaluations. And again, it's not limited to just labs or the diagnostic or therapeutic or prognostic world. It's for any sort of, you can use this for devices. You can use this for any sort of other medical or clinical, care or service that is being provided in order to see if the evidence threshold for them is at a level where they can be used for routine clinical care. They're answering them questions in order to establish their medical necessity. So let's go through this. P stands for patient population. You have to define your patient population. So in PICOTS, PICOTS, the P is for patient population. You have to define it. Sometimes you may have to get into subpopulation. This is where you define if you are concerned about certain age ranges, if you're concerned about gender, if you're concerned about maybe stage 3 versus stage 4 cancer, what type of cancer? Are you just concerned about the primary populations, or are you again, like I said, there's subpopulations that are involved? This is where this all goes back to questioning. This is where you establish your questions, and defining your patient population becomes extremely important, because that's going to drive all of the further questioning that you have. So paying attention to this is extremely important, goes without saying. I stands for intervention. It's whatever, like I said, these frameworks apply to not just testing, but for any sort of technologies that you are evaluating. Intervention is whatever you're introducing, whatever you're gathering evidence for, that you're going to bring to the picture of the clinical scenario, and it's going to make a difference. And this is where the claims that are being made about the intervention is extremely important. Just be careful if you're saying that the intervention is going to do X, Y, Z a priori. Your publication results in the peer-reviewed literature have to show that, because that's what is being claimed, and the rest of it is explained on the slide. So I won't get into that. C is the comparator, right? So how this is where the comparative studies become important. So, but I would like to take a pause here and sort of explain this further. A lot of times, what we see is that the comparative studies are being performed. They're being talked about. Oh, X does better than Y, right? And Y is the traditional way or the established way of doing things correctly. What I try to make sure I tell my audience all the time during my presentations is that make sure that X is actually medically necessary to begin with. Before you can compare X to Y, with Y being the established way of doing things or the traditional way of doing things, you have to first establish that X is achieving the clinical outcomes that it's stating that there's enough evidence for it. So before you get into comparative studies, first make sure that the technology, lab testing, or whatever your intervention is performing at a medically necessary level before you say before you state that that particular intervention is clinically superior to anything else that is currently available. And that includes, again, not doing anything, just monitoring or just monitoring. So just keep that in mind. I try to make sure I deliver that point during my talks. So the O, let's talk about the O in PICOTS, PICOTS, again, PICOTS. O stands for outcomes. Outcomes extremely important, goes without saying. And, this is where you need to establish your primary outcomes versus secondary outcomes and have the correct epidemiological principles, statistical analyses in order to establish the primary outcomes, in order to take a look at your data when you're doing your publications, because that's what is going to be looked at during the evidence evaluations for these. What was decided a priori, what sort of ad hoc analyses were performed, and what importance do they bring to the table, and can the ad hoc analysis, for example, be taken to establish significance or not. Another important point is to distinguish between a clinical significance and statistical significance. It may turn out that the clinical significance is of extreme importance, and if that is not established part of the situation, the statistical significance, achievement of statistical significance may not be enough in order to establish medical necessity. And then also, this is where the appropriate sample size is, or again, like I say, analyses that have been decided that are going to be performed with data becomes extremely important, where they correctly chosen. And then also, the quantity and quality. Is it that there is sufficient aggregate data to verify, the primary outcome results that are being discussed or that are being pitched as being enough in order to move forward with a medical necessity position, for instance? Or is the quality good enough? If what I talked about in some previous slides, if you're supposed to have well-designed controlled studies and even have introduction of randomization in your studies, and those are the only ways in order for you to establish for instance, clinical utility or establish medical necessity, or to come up with criteria for where your technology or testing would be appropriate. If the design is not good enough of the study or studies, going back to the pyramid, for example, that I discussed, then further testing is required because there's limitations in evidence. The biases that are present in the whatever the published evidence that is available at that time when the evaluation is being performed cannot be ignored because there's outstanding. So all these things come into play when you're looking at not just outcomes, but I wanted to make sure I gave you enough, again, buzzwords here for you to ask further questions if needed, or to go out and look at the references for further information that you need in order to make sure that the right quantity and quality of evidence is being produced and is being evaluated the correct way. T stands for timing. This is where the longitudinal study is the short-term outcomes good enough, or do you need long-term outcomes? Is 6 months good enough? Is 12-month data good enough, or do you really need to monitor for a couple of years, for instance? And again, this is all going to be defined by the exact questions or the intervention, and sort of what we talked about in the first 3 or 4 slides, what kind of testing intervention or technology we are talking about, but making sure that the timing part of it is important as well. Setting. This is also part of the PICOTS model and overall evaluative framework that is being used. So I think the slide explains itself. It becomes extremely important. It becomes relevant again depending on the study that you have. But again, just wanted to include this in here for you to be aware of it. And I think a large percentage of you, this may not be applicable, but you never know the situation you can turn into. But just having this knowledge that the setting, achievement of your outcome in the appropriate setting, if the setting is relevant, becomes extremely important too when evidence is being looked at. Again, situation dependent here. But this is towards the end of the presentation. Again, want to highlight what is it that we're trying to achieve here. Whatever trials, whatever the clinical evidence is out there, it should have high strength of evidence. This goes back to having the correct type of studies, the correct quantity and quality of clinical investigations that are peer-reviewed and published. Again, not anecdotal, not abstract, not posters. And this is the highlight of the bullet points, or the highlight of what they should be showcasing. They have a low risk of bias. I talked about bias in my presentation today. That as you go up that pyramid, biases are reduced. There's adequate power to address subgroups. We talked about population. I talked about how subpopulation, subgroups, you need to define them correctly. What does power mean? Enough sample size, big enough sample size. For your study or studies, aggregate is big enough, or your primary seminal work has a large enough sample size that is required in order for you to establish clinical validity or clinical utility. And then let's look at the second bullet: examine clinical strategies and complexities that are more likely to be replicated in practice. Remember, we talked about routine clinical care. Your data, your evidence has to show, and we talked about, any average patient walking in that meets the clinical context for which you are pitching your technology or testing for. The outcomes, the expected outcomes, need to be achieved similarly across the board for those average patients that are part of your population. And so that's where sort of the second bullet gets you to, that you should be, your results should be similar. There shouldn't be there should be reliability in them, and in order for you to become part of routine clinical care. The first bullet again goes back to our, patients, defining your population accurately, and because eventually when you do get data, you have to be able to apply them to the population. If there's too much variation with your population, or it's not being accounted for, that can be a limitation that future studies or investigations have to overcome if you are deciding to do further evidence development. And then, I wanted to just bring up a couple of things that we don't have time for today: direct-to-consumer testing, and then also pricing, involvement, and how that is achieved. Again, a future presentation from me, I would likely address these, but if you have any pressing questions that you want to bring in for these, more than welcome to share through LabRoots. And that concludes my presentation for today. I hope it was useful. And again, questions or any clarifications that are needed, I encourage you to reach out to me through LabRoots. Thank you for your time.
Thank you, Eugean, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. And as a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.